Wonderful. Tēnā koutou. Hello to everyone. So my name's Anna Santure. I'm based at Waipapa Tamataro, University of Auckland in Aotearoa, New Zealand. But I have the great privilege to be actually presenting today on work from my PhD student Kamopat, or A, as he calls himself. And Kamopat is handing in his PhD at 8 p.m. tonight, our time. It's super exciting. And this is the last wonderful chapter of what's been a fabulous body of work from his research. So I'll talk you through a wee bit of that. So, of course, to begin, a huge thanks and acknowledgement to Kamopat for leading this work. But I also wanted to shout out to the absolutely fabulous research fellow in our group, Katerina Stewart, who's really mentored Kamopat through lots of this analysis and has built the groundwork for the genomics project in Common Minor alongside Annabelle Whibley. We're thinking about invasive species. Obviously, invasive species have massive impacts globally. They impact biodiversity, they impact human health, and they can impact human livelihoods. And we know that the cost of both controlling and dealing with the repercussions of invasive species are around, say, $200 billion every year. But I'd argue that invasive species are actually really interesting models for us as evolutionary biologists. And in particular, we see for invasive species, they're invasive because they're good at going into new places. And so they go in a replicated manner into new environments, and we see very often very rapid adaptation to those novel environments. And so we can really start to think of some really interesting questions in an evolutionary context in terms of how does that rapid adaptation occur? Does it occur, for example, via standing variation that somehow survived through a bottleneck into the invasive range? Or is it via de novo variation, perhaps independently in each of these invasion lineages? So we set to look at this in a lovely model of invasion biology, the Common Minor. Oops, sorry, I double skipped there. Common minor are native to South and Southeast Asia, but are now pretty much globally distributed. They're present in every continent except for South America, hopefully never, and also in Antarctica. And they were actually introduced by humans in around the late 1800s as biological controls. So thinking about Australia and New Zealand, for instance, they were introduced by what we call our acclimatisation societies, these Europeans, English people predominantly, that were bringing species from outside New Zealand and Australia in to establish in these countries. And for Common Minor in particular, they were brought in as a result of large-scale herbivory on crops that were being planted as Europeans cleared traditional lands. So a real product of colonisation in terms of where they are now. So there are lots of ecological reasons why common minors are good invaders. So they are generalist, they're omnivorous, and they really thrive in habitat in human-modified environments. But we also see really good evidence of rapid adaptation in their invasive range, which includes adaptation to novel diets, morphological adaptation, as well as behavioural adaptations. So we really set out to ask about the genetic basis of this rapid adaptation, acknowledging that quite a lot of that might be some degree of plasticity, but really thinking about how we might use signals of selection in the genome to really help us tell that story of how they've adapted in the invasive ranges, and if so, if we can find those regions, what those regions in the genome might be up to. So invasive common minor, as we can see, are native to South and Southeast Asia, but actually all of the invasion lineages in the world are sourced from India. And we had this wonderful access to samples that were gathered, quite contemporary samples actually, that were gathered from India in the late 1970s and early 1980s that were held at the Royal Ontario Museum. And alongside those samples from India, there were samples from Royal Ontario Museum that actually covered South Africa, Fiji, some other Pacific islands, as well as Australia and New Zealand. But we also have modern day samples available from a lovely population genetics study from Australia, and then some very enthusiastic people controlling minors in their backyards in Aotearoa, New Zealand, that were happy to send us some bodies in the post. So basically, we combined all of this data from the slightly older museum samples with samples from Australian Museum covering the Australian range and our New Zealand samples, and we used a reduced representation sequencing approach, and A was able to trace the introduction history of common minor using that reduced representation sampling of the genome. So just a little snapshot of the genome that gives us an idea of overall population diversity and structure between populations. So A was able to reconstruct this really beautiful invasion history of common minor into its invasion ranges. And in particular, we identify a fairly distinct population in Maharashtra that we call Maharashtra subpopulation A. And that's actually been the source population for Fiji, but also into Australia. And actually, the birds that we have in Aotearoa, New Zealand, are actually from Australia itself. So it's a secondary introduction into New Zealand and two independent introductions, we think. And then from somewhere else in India that we don't seem to have sampled from these museum collections, there's been a completely independent introduction into South Africa. So taking that, that was the story from the reduced representation data that's actually very nicely corroborated by historical records such as shipping records for these birds, so it's always nice when that agrees. We actually selected between 8 and 10 individuals, 5 and 8 to 8 individuals to sequence at fairly good depth at a whole genome sequencing level to really start to ask, well, are there signatures of selection in the genome that might help us to explain this invasion success outside of India? So a beautiful work led by A and by Katerina Stewart, the research fellow in our group. They mapped, or Katerina actually helped annotate the common minor genome that Annabelle had assembled, and then she mapped these reads to the common minor genome, detected a lot of diversity there, so 9.5 million SNPs or polymorphic sites in the genome. And A conducted a number of scans of selection. The first two using this Baypass software, and Baypass software is really nice because it accounts for this population structure. So as I explained it, particularly New Zealand is this really interesting bottleneck but also very nested population within this invasion lineage. So really, really important to account for that structure before we try and detect these signals of selection. So we were looking at genetic differentiation using this XTX statistic, which is an FST-like statistic adjusting for structure, and A also looked at an allele frequency contrast between invasive and the native range, adjusting for population structure as well. Separately to that, he used the REHHR package to look for these haplotype signals of selection from haplotype homozygosity. So we know that an allele, when it's selected, will drag through a whole haplotype, a whole region of the genome with it. So we can see that signature of selection between the selected allele and kind of the ancestral allele. So onto his genome scan results. So just to orient us, on the y-axis is a support for selection effectively, so higher values give us more support for selection, and each of these alternating colours indicates the different chromosomes in this beautifully assembled common minor genome, and each dot is one of these SNP markers in the genome that we've tested for this signal of selection. And what we can see is that each of these metrics, the XTX on the top, and then the C2 allele frequency contrast in the middle, and this extended haplotype homozygosity, each have somewhat independent signals. And even though we'd adjusted for population structure, we were a little bit suspicious about any of these individually, but we were very, very confident in this shared outlier region. All three of these statistics are pointing us to this region of the genome that we term this key outlier region, and enabling the adaptation, this rapid adaptation of minor as they move from their native range into their invasive ranges. So it looks like a lovely, well-defined peak just there, but actually if we zoom into that region, it's sort of interesting. We actually see that peak of selection signal split a little bit. And it won't surprise too many of you to know that what the dip in the middle there is actually a structural variant. It's an insertion deletion, so some individuals have that region of the genome and some individuals do not. And so that explains that kind of dip in that support for selection through that region. If we look upstream of that region to the left, we can actually see that there are two annotated genes in that region. They're both actually copies of each other, so we've got copy 1 and copy 2 of AMY2A. It's an amylase gene, and amylase is really important in starch digestion. It's pointing to some sort of diet adaptation in this invasion process. We also see actually within the structural variant that there's a transposable element in there. We know that transposable elements can affect gene expression, and further downstream is a non-coding RNA element, again something that could influence expression of potentially one of these copies of the amylase gene. So who is the suspicious point of selection then? So what A did was to actually look at the top SNPs that were showing these signals of its selection either side of the structural variant. And I'm sorry, it's very small text and I can't even see it properly on my screen. But I think the very first column is the IHS, so the extended haplotype homozygosity peak, left hand of this SV. And then we've got the XTX, the population differentiation peak, and then the C2 peak. And then the structural variant itself, and then downstream of the structural variant, we've got a combined SNP that's the top peak for two of those statistics, and then a C2 peak a wee bit further down. But what I'm just showing here is that each line is actually an individual, and they're organised by their populations. And the single colour that you predominantly see in the top means that all individuals are homozygote through most of those SNPs. So what we can see is this really interesting split that all of the invasion range is basically homozygote at these key SNPs showing signals of selection. Whereas in the native range, we see this really interesting polymorphism that's present. We see all three colours representing the three genotypes at those positions. So clear polymorphism in the native range that seems to now have been fixed as we go into these invasive range samples. And in particular, just remembering that the very top population there, South Africa, is from a completely independent source in India. So we're seeing this signature of parallel adaptation. Now, what I glossed over a little bit was that the third column there, our C2 peak, this showing this very strong allele frequency differentiation between invasive range and native range, is actually a missense mutation. And it's a missense mutation in the second copy of AMY2A. And it's not just any old little mutation, thank you. It's actually a charge and size differing amino acid change that changes from a proline to a histidine. So we suspected this might be a fairly big thing for the protein to be coping with. And indeed, when we look across other passerine birds, every other passerine bird that we looked at has a C at that position, indicating the ancestral state. In this AMY copy 2, in common minor, we actually have an A, and our reference genome, which is the top line there, is a bird from the invasion range. So they've got the A there, and everyone else is looking different because they've had fixed this new variant. And if we look at AMY copy 2, AMY2A copy 1, upstream in the genome, that's also got that ancestral variant. So really showing that we've got this amazing fixation of this derived variant in the invasive populations in the second copy. AMY2A has also been implicated in adaptation, rapid adaptation in other species. We see that it's associated with human commensalism in house sparrows, and genes in that family have also been associated with rapid adaptation to high-starch diets in human and dogs. So just to finish up, I think we've been incredibly fortunate to be able to place context on the samples that we had from New Zealand and Australia from this incredible record from the Museum of Royal Ontario Museum that have actually enabled us to say that we can see parallel evolution on the standing variation across the invasive range. A smoking gun is obviously that non-conservative missense SNP in copy 2 that might be messing up or changing at least the protein function, but we can also might be influencing protein expression from nearby things in the genome, such as that structural variant in the non-coding RNA. And hopefully overall we've shown that replicated analyses within and across species can really help us understand rapid adaptation at a molecular level. So thank you very, very much. And if I've got one moment left, I welcome any questions or comments, but otherwise please grab me afterwards. Thank you. I think I've got a microsecond, maybe not. Yeah. No, we haven't actually, no. So yeah, it would be lovely to look more generally, and we know that there are actually other populations, I think, such as in, I can't quite remember, maybe in Mauritius that have actually probably come from not not from India. So it would be lovely to sample both of those. Yeah, thank you. Cool. Thank you.
Yes, bonjour, hi. I'm Jeremy Yoder from Cal State Northridge in Los Angeles, and I'm going to tell you about some work that we've almost got published now, reconstructing 120 years of flowering events in my favorite spiky desert plants. So these are Joshua trees. They are the biggest member of the plant community. Most of the places they grow in the Mojave Desert. They're important for all sorts of cute wildlife. And of course, they're maybe best known for having a very specialized pollination relationship with yucca moths. We've done a lot of previous work describing Joshua trees' differentiation in association with two different pollinator species. There's an eastern Joshua tree and a western Joshua tree that are essentially parapatric across the desert. But another thing that people have done with these trees quite a bit is because they are so apparent on the landscape, they're really good for species distribution modeling. So this is, of course, the class of methods we all use frequently, where we have presence and absence information about a species on the landscape. We correlate that with, or connect that to environmental conditions, feed that into some kind of modeling framework, and use that to identify relationships between presence on the landscape and the environmental conditions so we can predict where the species is. For Joshua tree, that's been especially relevant for projecting range changes with climate change. But there's lots of other applications. We have a paper comparing the eastern and western Joshua trees' climate niches and showing that they use pretty similar conditions. But the information in these models, and this is not a new observation, is not very rich. It doesn't tell us much about what's going on the ground, right? A species presence could be one dying individual. It could be a happy growing population. And this is also really evident for Joshua trees, right? Here's some Joshua trees in Walker Pass, almost on the Pacific Crest Trail. Very happy population, having a really good flowering year. And a couple hours drive away in the lower parts of the Mojave, same year, basically no flowering going on. Still a relatively happy population, but very different density. And both of these would be presences in a standard model. What we can do though with these trees also is, because of the way they bear flowers, we actually start to have an index of what the condition of the population is, how they are doing in a given year. So I realized this looking through records for iNaturalist, which we've, I think, already seen mentioned. Joshua tree is really well represented in iNaturalist. It's a rich source of data, and it's possible to go through and spot the condition of individual trees in the records that are uploaded through the app. So a couple years back, I talked a very hardworking student, Ana Karina Andrade, into working through the Joshua tree records that are on iNaturalist and annotating them for the phenology, or the phenophase that she saw. And we identified and validated over 10,000 records from 2008, when the app launched, up to 2022, which is the latest data we have for this. So these are of course very heterogeneously sampled in time and space. You can see there's been a dramatic increase in contributions over the years that reflects the expanding user base of iNaturalist. And if we grid these out to a 4-kilometer grid and distributed them on the map, there's some real clear lack of sampling in parts of the Mojave that are less accessible. That really big gap north and west of Las Vegas, that's the National Nuclear Testing Range. There are not a lot of tourists looking at Joshua trees there. But we're covering a fair bit of environmental variation here, and we're covering a fair bit of time. And importantly, the sampling heterogeneity in time, that increased sampling density in recent years, doesn't translate into differences in flowering frequency. We're not seeing a confounding trend in the incidence of evidence of flowering. So it occurred to me, or the idea behind this is that we can take this annualized data and relate it to weather conditions leading up to each flowering year and actually start to model the conditions that prompt flowering. So it looks more or less like this. We have an observation of flowering or not at a given point in time, and we place that in a flowering year. And imagine working backwards. These are long-lived trees. We expect that, like other desert perennials, they may be responding to signals about the conditions they're in over multiple years. So I linked records with the PRISM database, which covers total precipitation, maximum and minimum temperatures, and the vapor pressure deficits, index of drought stress for plants on the ground. And also looked at differences from year to year, which is going to turn out to be important in all of these. And the framework that we used to model this is something called Bayesian additive regression trees. This is a larger set of the workflow, but basically I organized the records by year and by binary flowering status, so anything with buds, flowers, or fruits just gets binned as evidence of flowering at a given time and place. Aggregate that to the common raster grid with the weather data, and model the presence or absence of flowers as in relation to various possible environmental predictors from the PRISM data set. So BARTs are a really nice machine learning framework. They're very flexible. They're pretty computationally efficient. And they let us do some stuff that help to anticipate potential issues with the data, but also see what the model was doing when we had finished training it, which is handy. And among other things, there are some really nice utilities in the Embarcadero package developed by Colin Carlson, who is an important co-author on all of this, including this funny-looking graph here. This is predictor selection in Embarcadero via BARTs. And this essentially fits BART models of varying complexity, varying numbers of decision trees inside the model package, and uses how frequently a predictor is included in the tree, in the model, as an index of how important it is. So very simple models need to be selective about what predictors they use. Very complex models with lots of trees can just pull everything in. And essentially what this does is it gives the model, the predictors that are important end up over here where they are showing up as high importance in the simple models with few trees, and as you get to those big models with 200 trees, everything is just sort of coming out in the wash because they're complex enough they handle it all. So here are the posterior modeled effects of the top 6 predictors for Joshua tree flowering. And the final model has an AUC of about 8.84, which is decent for differentiating flowers versus not flowers. But more importantly, these partial effects are biologically realistic. So just to interpret these, because the precipitation contrasts are a little bit hard to wrap your head around, this basically says that flowering is more likely when a wet year follows a drier year, when winter temperatures are comparatively mild, you've got a peak in the marginal probability above 0. When maximum VPD, vapor pressure deficit, drought stress, is relatively low, and when VPD and temperatures have been comparatively stable over the last couple of years. There hasn't been a big swing one way or the other. We can also, with Embarcadero utilities, generate what are called spartials, which is just the partial effects projected in space. And this is pretty neat because you can actually see how the conditions in different parts of the desert, different parts of the range, are contributing to higher or lower probability of flowering. So this is the original data from 2019, which was a good flowering year. You can see we've got a lot of positive presence of flowers. And you can see how the precipitation predictors in particular are lighting up blue there. In 2020, which was a bad flowering year, those same predictors are indicating much lower probability of flowering. But there's variation in space, and that's part of what we're also getting in the model. So with a fitted model then, and this is the cool thing, we have weather records going back in the PRISM database to before 1900, right? So I can take the trained model and generate predictions for the entire range of Joshua Tree all the way back to the early 20th century. And so the color ramp here is indicating higher probability. The best power classification cutoff is relatively low. That's determined in another procedure that's available in Embarcadero. And with that hindcast, so it looks good, but we wanted to dummy check it. So I went looking for basically any available independent information. We had collaborators who have field notes that they've taken, but also, of course, and this is the most relevant for this symposium, museum collections or herbarium collections, the National Phenology Network records, more formal records of flowering. And also, Joshua Tree blooming events not infrequently make the news. This is a 1924 LA Times article by they called them Cadillac tourists, who saw a bunch of Joshua trees with fruit on them and told a reporter. So I can take these and relate them to what the model says for the location and year and see how it lines up. And it's pretty good. So here's the predicted probability of flowers being present from the model output versus whether flowers were recorded or not in herbarium records, formal sheets, the NPN data set, and so the median for true is above the classification cutoff. There's also just a straightforward t-test that's a highly significant difference. And for the news articles, which are handled a little bit differently, we also get a nice clear differentiation. That's the 1924 LA Times article. There's a 1961 account of poor flowering. So with a hindcast that we have some confidence in, I can start to think about what has been the history of Joshua Tree's experiences in different parts of the Mojave, and how does that relate to the conditions on the ground, right? So this is now an enriched sort of species distribution model output. And what we can see, first off, range-wide, we get a median frequency in flowering of about 1 in 4 years, which aligns more or less with my experiences planning field work. And we can think about change over time, right? So this is now change in the number of flowering years for the first 3 decades of the 20th century versus the most recent 3-decade period in the study period. And so we go now from a median frequency in the early 20th century, estimated about 1 in 5 years, to a median frequency in the recent period of about 1 in 4 years. So it's actually, there's been a slight increase in conditions supporting flowering. What's going on there? Well, all of these changes, right, are just arising from trends in the PRISM predictors. And those changes are not necessarily better for overall recruitment. And I'm almost out of time. But thank you all. And this is going to be in Ecology Letters as soon as they release it.
All right. Hello, everybody. Thanks for staying around. I'm Tais Ribeiro. I'm a PhD student at the University of Maryland. And I'm here to talk about my research on biogeography and how climate influences the evolution of an oil bee genus. And yeah, and I'm still working on it, but I'm looking for postdocs next year. So in my research, I am looking at how different factors can drive the evolution. So I'm looking specifically at biogeography, climatic variation, and interspecific interactions. And interspecific interactions can act as selective pressures, influencing how adapted the species are to their communities. The biogeography, history of the areas that the species are, can also lead to evolutionary pressures, as well as the climatic variation can lead species to evolve and adapt to, to different climatic niches, which will eventually change evolutionary patterns. So in my research, I am looking at bees from the genus Calipogenus and using them as a model to understand how all these factors can play a role in evolution. So Calipogenus bees are oil collecting. So that means that they go to specific flowers and collect floral oils. And they have specialized pollinator interactions with these flowers that they collect oils, even though they do collect pollen and nectar in other flowers. And they use these floral oils to feed their larvae and waterproof their brood cells, which is an interesting thing that not all bees do. Some specific bees have this behavior. And they are South American. And they're in the tribe pt.2, and all of these species in this tribe are also oil collecting and also in South America. And there are 29 known species of Calipogenus, which were divided in 7 morphological groups, which I will refer to later. And in my research, I am reconstructing the phylogeny of this genus, and I have used ultraconserved elements, and this tree is the Bayesian time tree using the 200 most informative loci. And the different colors mean the different morphological groups that I mentioned before. And I have used a large sampling of Calipogenus using most of the species, using 17 out of the 21 described species plus 7 undescribed species. And in this tree, I have 108 of my total 114 specimens. And I just wanted to note that most of these specimens come from museum species. So 64% of my sampling come from museum species, either land museum species. And this is the same tree, just collapsed by 1 species per terminal, so that is easier to look into. And also this is this tree that I used for analysis that I'll show later. And in this tree, what we see is that the origin of the group was around 30 million years ago. And we see that the morphological groups are monophyletic. However, the genus is paraphyletic, so there's still things to be seen in there, and it's mainly in 2 different clades. So we see that from that node in 30 million years, with the origin around 25 to 20 million years ago, we see first this clade that includes those 2 groups in purple, the group Geodianus and Parvus, sister to a different genus in Tapinotaspidini, and we have proposed a separate a change in the genus of this species already. And in the different clade, we see the group, this is group Millery, the one that you probably can see in the middle of the blues. This is the type species, so we see the type species is there, together with several new undescribed species. And sister to that, there is a clade with all the other species, but sister to yet another Tapinotaspidini genus. So we see that the genus is paraphyletic, and then there's still taxonomic revision that would be good to do in this group that I'm doing part of the phylogeny. And but now that I have this time tree, I can use it to ask and to look into the evolutionary questions. And in this talk, I'm talking about the biogeography and the climatic variation, not about the interspecific interactions with the flowers that they visit. But if you're interested, come and ask me questions after. So I'm gonna end my questions in this talk are: what is the biogeographic history of the group, and what is the role of climate in its evolution? And I'm going to walk you through this by first telling you some of what is the data that I use, and then the analysis that I did, and then the results for each of those questions. So for the data sampling, I have done a literature and collections review to identify the ranges and the climatic preferences of Calipogenus. So I have compiled georeference observations from natural history museums, either by directly contacting them or accessing digital databases such as GBIF, and literature review, also reviewing papers that have collected Calipogenus. And this is how this is the data that I have. So I have a total of 815 unique observations of Calipogenus, and again, most of those come from museum specimens. And in this map, again, the different colors are the different morphological groups. So we see here that the groups are structured geographically, and we could recover those ranges in there. And we see that there are three main groups that are Andean, so the orange and red groups are Andean, and the other ones are more associated with grasslands in South America. And for identifying the climatic preferences, I have combined those georeference observations with bioclimatic variables from CHELSA. And then now let's look at the analysis. So first for the first question on the biogeography, for the analysis, I use a prudent time tree, so I remove some of the outgroups for this analysis. And the areas that I used 7 areas based on the ecoregions from WWF, based on the biomes and different combinations. And I have used biogeobears, and I have tried all the models that they have available, DEC, the Bay Area-like, and Diva-like, with and without the jump dispersal J parameter, and to see which one would be the best model from those. So the best model for this data was Bay Area-like with log likelihood of -118. And I'm going to walk you through these results. So the first thing that we see here is that the origin, we recover the ancestral range of these genus in an area of grasslands and forests, Atlantic forest and Andean forest. And we see that this was maintained until around 20 million years ago. We still see this range with this combined area. And then after that, we can see both clades. So first looking at that first clade, the one that we have the separate genera. So after this 20 million years ago and staying there, we see a range expansion to the south, to the temperate grasslands, and a little to the central Andes too, and with some later shifts happening within the species in there. And now let's look at the other clade. Again, they were in this combined area, and then from there, around 20 million years ago, there was the origin of the central Andes. So this uplift could have influenced the evolution of these groups. So after this uplift, we see that the groups either would expand into, we see either an expansion into the Andes, or we see a speciation in situ in the grasslands and part of the area of the Atlantic forest. And so the group that has the type species, group Millery, we see that this group stayed in the area of grasslands and Atlantic forest, and the new species are all in the grasslands in the diagonal of open formations in Brazil. So they are in the Cerrado and Caatinga in Brazil. And the other group expanded to the Andes. We see first this expansion and then followed by isolation in the Andes. And then the different groups will either stay on this area of the central Andes and the Andean forest, or we see an expansion to the south with the temperate grasslands and the South Andes. So take home from that part of the biogeography, we see that there was an origin in the grasslands and forest area, and later a dispersal to the Andes. And this, one important thing to mention is that the timing of these events is congruent with the geological history of South America. But now let's look at the climate. So looking at the road of the climate, I've used a new method called BFINE. And with this method, we have reconstructed the climatic niches informed by phylogeny. And to do that, we use the time tree and the occurrence data, the time tree with all the outgroups and the occurrence data for all those species, including presence and generated pseudo-absence for it. And I have used the bioclimatic variables I had used the mean annual air temperature and the annual precipitation. And this is how the results look like. So these are the ridge plots. So what we see here is we see first the phylogeny, and then the curves are the climatic niches for the species reconstructed using the phylogeny. And we see first temperature and then is precipitation. And this center is the climatic optimum. What we see here, again, with those colors for the morphological groups, we see that there is clustering with the clades. So we see that the niches are congruent with the clades that we reconstruct here. And there is a negative correlation between the niche optimum and the breadth. So for example, in the species that are adapted to hotter climate, they have a narrower niche breadth, so there's not a lot of variation in there. And one interesting thing that we notice here is the Andean species, so those orange and red on the top over there, we see a shift there and to lower temperature and precipitation. And there's kind of like a faint yellow line in there that you can kind of see how like there is a shift from there to from the other species. So we see here with the role in the climate, we see how the Andean clades are adapted to colder temperature, which could be related also to what we see in the biogeographic history and the adaptation to the Andes. So another take-home message from this research is that it wouldn't be possible to do this evolutionary conclusions and to do this whole work without having access to museum collections, especially contacting collections in South America where these species occur. And yeah, and natural history, and access to museum collections and natural history data. I'm also using natural history data in other parts of my research in here. And with that, I would like to acknowledge the Spindle Lab, the Weather Lab for having me visit while I was working on the climate, and the museums that lent us, and the people that lent us specimens. And thank you for the Spozio organizers for inviting me, Funny Angels Days, and thank you everybody for listening. Yay. And I guess I have time for questions. Is anybody? Yes. Yes.
Lovely talk, Vicky. I was wondering, so you showed that some of the hot adapted species are already in a fairly narrow climate niche. Do you have any inference about what can happen with those species in particular, climate change, or whether they're more or less, how the different species might respond to climate change?
Yeah. So. These species, like we see them more like in the south of the Andes, so they are in grasslands that are like more, colder. So we could see that maybe like with the expansion of the grasslands, and then maybe with like climate getting like with the weather getting hotter, they go like up in the mountains to like colder temperatures in there. So maybe that could be like what was happening in there. So it's more like in a historic manner. So yeah, it's associated with several events in South America, like with expansion of grasslands also, like in colder climates, and then the retraction, and then the expansion of tropical forests. They're more associated with grasslands and open formations and stuff.